So a presentation
Okay.
of Life in the Dark, the microbiomes of mussels from deep sea.
So we're going from outer space to the deep sea. Our research in the symbiosis department focuses on chemosynthetic symbioses, and you see here that we study them from a wide range of approaches, really a holistic attempt to understand how these symbioses function. And the way chemosynthesis functions is chemosynthetic bacteria. We already heard a little bit about them. They use energy sources such as sulfide or hydrogen or methane and carbon sources, and they use the energy when they're oxidized, these compounds, mainly through oxygen in these symbioses. And harness that energy to fix CO2 into biomass and live in symbiosis with animals that use them as nutrition and then eat them. Remarkably, although they were discovered in the deep sea in 3,000 meters water depth, we now know that these symbioses are widespread in many environments, including shallow water environments, right off of many coastal environments. And they've evolved numerous times, these symbioses. You see a phylogenetic tree or phylogenomic tree here, where all the colored lineages are bacteria that have evolved chemosynthetic symbioses with hosts. They're often closely related to shallow water bacteria, and there's a wide range of bacteria, 15 different bacterial groups that have evolved these symbioses, and 2 protists and 9 animal groups. And what that tells us is when we see something happening repeatedly in evolution and convergence, evolution, that there's a strong selective advantage, both for the bacteria and the animals, in coming together in these associations. We work on the deep sea mussels that I'll be telling you more about in my talk today, but also on the shallow water hosts that you see here. And we use a wide range of methods to work on them because the microbes can't be cultivated and it's extremely difficult to keep the hosts in the lab for longer time periods, not to mention getting them to reproduce. So we go from the full range of omics to lots of different imaging methods and incubations in our field work. So let me tell you about deep sea mussels and how they work. What you're looking at here is a micro CT image of a Bathymedialis mussel, and the symbiotic organs are the gills. They contain the symbiotic bacteria. They're very greatly enlarged because they house so many bacteria. We estimate that an 8 centimeter large mussel houses about 10 to the 12th bacteria cells, and they only have 1 or 2 symbiotic species. This means examining their microbiomes is relatively easy because we're not dealing with a great diversity of species that, for example, make studying the human gut microbiome so challenging. And the symbiotic bacteria sit inside the host cells. What you're looking at is a fluorescent in situ hybridization image of a gill filament. And what you can see here in the red box is a single bacteriocyte, a gill cell, in which the symbiotic bacteria, sulfur oxidizers in yellow, and in magenta, the methane oxidizers, are sitting in this epithelial cell. And you can also see that not all cells are colonized. The host, the outer tissues that are full of cilia and mitochondria, the host doesn't allow the symbionts to colonize these. Only certain gill tissue cells are allowed to be colonized. And these are, as I emphasize, epithelial tissues, similar to intestinal epithelium. So when you think about a gut, then what you can think about in these gill tissues is more like an inside outlining, with the gill lumen in the middle here, where you see the macrophages patrolling, or hemocytes patrolling the gill tissues. And this is the region in which the symbiotic bacteria sit, and they're eaten through phagolysis, lysosomal digestion. But today, I'd actually like to tell you about a parasite, not a symbiont. These are some of our newest results that we recently published and that we find quite intriguing, and I hope you do too. So theory would predict that when you have these types of dense communities of any type of animal, we know that whether it's bird flu or in agriculture, or when we keep animals, is that they're an oasis for parasites. And we find remarkably few parasites, but the one that we did find is quite intriguing. And the two people that did the work are Nicolaish, who is now the head at EMBL, the head of Mobile Lab Services, and Miguel Gonzalez Porras, who did a postdoc at the MPI for Biology in Tübingen, is now at the University of Malaga. So the parasite that we found, a gamma proteobacterium, I'll show you a tree in the next slide, a single pathogenic microbe, Endonucleobacter, infects the nuclei of the deep sea mussels, and it replicates. You can see here it extends, then it divides longitudinally, and then it starts to septate. And in a single nucleus, up to 80,000 bacteria then grow from the single bacterium that infected it. And this greatly balloons the volume of the nucleus up to 50-fold. And at the end of the infection cycle, then what happens is that the nucleus and the cell burst and the pathogens are released and the infection cycle begins again. And as I mentioned, they're related to shallow water bacteria. What you see here is a phylogenomic tree. Up here is Endonucleobacter. And it's intriguing that they're related to a shallow water symbiont called Endozooic communis that we like to call the E. coli of the oceans. It's been found in quite high abundances, for example, in corals. And people, there's a nice paper that came out about Endozooic communis, which is frenemies of the reef. It's unclear for the Endozooic communis if they're actually pathogens or commensalates or symbiotic bacteria. For the pathogen that we're looking at here, the related one, we can say it's definitely pathogenic. So only very few bacteria are known to infect the nuclei of cells, of animal cells. And the reason for that is you're hijacking the command center of the cell, so that you really, if you want to multiply, that's not a good place to be. On the other hand, if you want to hide from the immune system of an animal cell, it can be a great place to hide. So how does the mussel protect itself? Number one, the pathogen can only infect symbiont-free cells. So there are a couple of reasons we think it can only infect these, but we do not see it infecting any of the cells that contain symbionts. And here you see again a fluorescent in situ hybridization image of the, in green and red, the sulfur oxidizing symbionts, and where the pathogen is, there are no symbiotic bacteria. So what Miguel did was use laser dissection microscopy to look at non-infected nuclei of the mussel, then at early, mid, and late infection stages of the mussel to get a better idea about their metabolism during the infection, using transcriptomics then to analyze expressed genes. And in contrast to what we had first assumed, that they might be eating the DNA or the RNA of the host, we found no genes that were encoded for gaining nutrition for chromatin or RNA or DNA. And that makes sense when you think about it, because you want to let the cell actually be intact for as long as possible. And we see that the host cells remain transcriptionally and metabolically active. What it does is it gains its nutrition, the pathogen, from host sugars, lipids, amino acids, and possibly mucin, which is mucus on the outside, which is interesting because it's not quite clear how it accesses this. But we have some hypotheses about that as well. And it's clear that when you do the calculations of 80,000 cells, that the host cell it infects can't provide enough nutrition on its own. The resources, the host sugars, lipids, amino acids are drawn from neighboring cells as well. We finally asked, how does the pathogen avoid apoptosis? Apoptosis is programmed cell death, and that's one of the first responses that animals have when they're infected by viruses or by bacteria, by pathogens, is that they go into programmed cell death. We were very surprised to find that Endonucleobacter expresses as many as 15 inhibitors of apoptosis. And the reason we were surprised is because these proteins, these genes, are only known from animals. And they're not known from any prokaryotes. They've only been found in a few animal viruses from insects where the viruses have acquired them through horizontal gene transfer. What inhibitors of apoptosis do is exactly as the name suggests. They inhibit apoptosis. It's closely regulated in animal cells because obviously they don't want to start this cascade unless they have to. And the inhibitors inhibit caspases, which initiate apoptosis from beginning the apoptosis. And so what we see is a real arms race between the pathogen and the host in initiating apoptosis. What you see here on the left side are the expression patterns or the expression processes that we saw of the inhibitors of apoptosis. Black means not expressed. Only 3 are expressed in the beginning stages. Caspases, when it gets red, are not highly expressed. And when we get to the late infection stages, all 7 inhibitors of apoptosis here are being expressed. And you can see the host is ramping up its caspases to try to counter this. Finally, we were interested in, well, if this is an animal protein, where did the pathogens get it from? And in a phylogenetic tree of the inhibitors of apoptosis, what we can see is that those of the pathogen are most closely related to those of its muscle host. So you see here in light blue all the mollusks, that bivalves and other organisms that have the inhibitors, and you can see that in dark blue the pathogen inhibitors are most closely related to those from their muscle host. Intriguingly, we have a Russian doll system here and evidence for the fact that the pathogen can be infected by viruses. Here in a TEM image, you see the nucleus, you see the bacteria inside the nucleus, and you can see capsids that are typical of DNA viruses that are infecting the pathogen within the nucleus. And so our hypothesis is that the pathogen acquired these inhibitors from their muscle host. That's clear from the tree. This is surprising because horizontal gene transfer from eukaryotes to bacteria is rare. Eukaryotes, as a rule, don't have a lot to offer in terms of their metabolism to bacteria. It's more the other way around. But what eukaryotes can offer to pathogens is ways to protect themselves against their immune systems. And we are hypothesizing that viruses might be the mediators of this horizontal gene transfer between bacteria and eukaryotes. And as is known also from the human gut microbiome and other environments like biofilms, physical proximity of microorganisms promotes horizontal gene transfer. And that's certainly something that we're seeing here. And with that, I'll finish up by highlighting the fantastic work that my group has been doing and the many sources of funding that we have. And thank you for your attention.
Thank you, Nicole, for your interesting talk. We will have time for question and discussion with Nicole at the end of all the 4 presentations. You can post question on the comments. Nicole Lab also works on the diversity, ecology, and evolution of symbiosis between microorganisms and different marine invertebrates from different environments, such as deep sea hydrothermal vents, cold seeps, and shallow water coral reefs, and seagrass meadows. So they use different methods from single gene analysis to omics and in situ experimental approaches. With these approaches, they showed different beneficial interaction with microorganisms, allowing the adaptation of these animals in different nutrient-poor environments. Thank you, Nicole. Our next speaker is Dr. Henk Borluis from Royal Netherlands Institute for Sea Research, and Hank will present how square microbial life in solar salters.
Okay, thank you. Am I audible? You can hear me, you can see my
Yes,
presentation. Good, good,
yes.
good, good. I will start. Okay, so how to square microbial life in solar salters. This is a small introduction to hypersaline life, life at high salt concentrations. But first, let us go a little bit to what really is salt, what is really hypersaline. And I think a good example is what I got when I was invited to go to a lake on the island where I work on Texel. And the newspapers briefed about this lake that turned completely pink and was hypersaline, according to the local ecologist. But when I went there, we measured the salt concentration. It was about 40 grams per liter, which is a little bit more than seawater. So yes, it's hypersaline, but only from the perspective of a aquatic freshwater microbiologist. For me, that's absolutely not hypersaline. So what I do work on more is like the salt lakes in Gironde in France, where you have these solar salter systems where salt is being harvested for commercial and industrial use. And you have kind of a microbial mats growing in these very shallow salt lakes. And you have all these fancy ring structures called fairy rings. So there you have about 150 to 200 grams salt per liter. But even more salt is my main topic would be the salt lakes, the solar saltons, and the crystallizer ponds that you find in Santa Pola. I've also done some work on deep sea brines, and you find also these very high concentrations in the brines that the astronomers think are present on the Jupiter moon of Ganymede. So these salt lakes are mostly commercial, but you also have some natural salt lakes. They have this beautiful coloration from algae, the cyanobacteria, green algae, and then at one moment the lakes start to turn more orange, more pink, and that is because of a lot of organism that has a lot of beta carotene accumulated, mostly as a protection against the high sun irradiance and the UV irradiance. Now, on this picture from Terry McGinnityk, you can see very clearly the kind of the holy trinity. Here we have three domains of life. We have the eukaryotic algae, we have the bacteria like Salinibacter, but we also have this very weird looking square organism, which turned out to be an archaean. Now, this square was discovered already in the late 1970s, published in 1980 by Tony Walsby. At that time, for a long time, named square bacterium because they didn't know yet whether it was an archaean. That was still a developing issue. And it has a very peculiar shape. It's like a post stamp. It's very regular angles, but it's flat. It's absolutely flat. It's membrane on membrane. One of the problems in further characterizing this was that it's unculturable. For a long time, at least until I came along, it was unculturable. And what is even more frustrating is that it's also the most abundant species in these brines. So you have a sample with 50% of these organisms are these squares. You go to the lab, you try to isolate, and you isolate everything except the square. Everything grows well in the lab except the square. So in the end, it took me about 2 years. So this is the moment where we started sampling the brines. You can see here is the remnant of the salt that's just being extracted for chemical use. And here, this is my friend and colleague Paco Rodriguez Valera from the University Miguel Hernandez in Alicante. And here we sample a small puddle of the hypersaline brine, almost a bittern with very high concentrations of sodium chloride and magnesium chloride. And it took me about 2 years growing on very low nutrient concentrations, low carbon concentrations. And finally, I had this organism into pure culture. Surprisingly, there was also a second isolate, was much more or less in the same period, which was isolated from a salton in Spain. I know in Australia. So you can see here these nice electron microscopy images that they really have perfectly square form. All these white dots that you see are gas vesicles. They are filled with air, whatever gas is around them, and it helps them to float around in the brines and preferably be close to the surface because they require oxygen. They are aerobic archaea. I named it Haloquadratum wolfsby. It was well published already in 2004. So we also started to sequence the organism, and well, what could have been done now easily for about 100 euros, we had to do in the beginning of 2004, 2003, for over 100,000 euros. You can't imagine it anymore. This was the cost to sequence about 3 million base pairs and a small plasmid of 30,000 base pairs. But at least we had the genome, published it well, and we can dive further into the ecology of the organism. Now, I have different interests in my long career, and one of them are the daily and seasonal rhythms. And this is an example of a rhythmic movement of sulfur oxidizing bacteria in a coastal microbial mat. So low salt concentration. This is a stop motion movie. And what you see is the movement up and down with the availability of oxygen provided by the cyanobacteria. Cyanobacteria have a true clock. They have a circadian clock. They do photosynthesis during the day, producing oxygen. But at night, or in the dark, they start fixing nitrogen. And that is a process that cannot tolerate oxygen. So this is why they separate these things. Very interesting, clear example of a rhythm in these examples, or in these samples. Now, when we go back to these fairy rings growing in Gironde at 150 gram salts, so this is a very shallow pond. There's only about 5 to 10 centimeters of brine above these microbial moss. They're not dry; they're in the water. And I hope I can show. So they grow very slowly, but every day a new ring is formed. If I shield it from the sun, there's no ring being formed. So it's a photosynthesis depending process. The layers are like a mucus layer, and it's full of dead diatom casing. We do not yet know clearly what the process here is, but very interesting. So the question is there any rhythms in Haloquadratum, other halophilic archaea? And the reason is because Haloquadratum is a phototrophic organism. It doesn't do photosynthesis, but has a bacteriorhodopsin that produces upon the incidence of light, captures photons, and the chromophore changes conformation, and protons are being pumped over the membrane. And this proton gradient can then be used to generate ATP via an ATPase. So it would be logical to spend as minimal as possible effort and energy, nutrients into making this protein. So, and in doing that, if you would expect that the gene is only transcribed closely before when it's needed. So you would expect kind of a circadian pattern in gene transcription to make this to produce this protein, and maybe also other proteins. So we did the transcriptome analysis with a cultured organism, and we compared the dark transcriptome with a night transcriptome, and unfortunately there was no difference. In the end, a negative result is also a positive result. These are the facts. There's no differences in our culture in the expression of any gene between light and dark. So, but we also were interested in seasonal differences. So we took samples from a winter and a summer from the same brines from which the Haloquadratum was originally isolated, but a couple of years later. And we again did a differential gene expression comparison. But there we saw a clear overexpression of a number of genes, including the bacteriorhodopsin during winter, relative to the summer. And most of these genes are involved in the energy maintenance. So during the winter, there's less sun, lower temperatures, and the species Halicodratum puts in more effort to compensate for this energy low period of time. What we also see, for example, is if we compare the gene expression patterns, these are the transcripts per billion, and this is how many genes. You see a kind of a bell-shaped curve for the two natural transcriptomes from a natural ecosystem. But in comparison, the cultured strain shows a completely different pattern of gene expression. So most of the genes are actually low expressed, and only a few genes are very high expressed. And in nature, we see this bell-shaped curve with more average of gene expression patterns. And we think that is caused by the fact that our culture is only one species, but in the natural environment, you have many different ecotypes of the same species. Now, we came aware of these ecotypes by doing this kind of a read mapping of a metagenome to the sequenced genome. And then we see these genomic islands, regions that are not well covered by the reads from the environment. And these regions are therefore unique to the sequenced genome. And that led us to the idea that, okay, there are probably different ecotypes. They are still the same species, but they have a different genetic makeup. Now, one thing has to be said is that, okay, these samples were taken about 7 years after the sample that I took for isolating Halicodratum, this strain HBSQ001. So I went back. I didn't went back. I went back to the freezer. The fact was that what I did, I wasn't planning to isolate Halicodratum because actually I didn't even know about its existence at that moment. But we took samples for DNA extraction and doing metagenome analysis. But the isolation became a success, and the DNA stayed in the freezer for a long time. And in 2023, beginning 2024, I gained a lot of money finally for sequencing these samples. And so we started doing the sequence, but we find more or less the same. We find still these regions which are very low covered by sequencing reads. Still means that the original strain is less than 2% of the total ecotype diversity at the moment of isolation. So we can wonder, did I isolate this species because it evolved in the lab during this 2-year period, or were these genetic differences maybe key for being able to isolate it? So one of the reasons why Halicodratum is successful at these high salt concentrations is probably the presence of this very large halomucin, a strain that Nicole just also, or the protein that Nicole also just mentioned. It was at that time the largest archaeo protein, more than 27,000 nucleotides, 9,000 amino acids. And it has all these kind of repeat sequences and a signal sequence, which is typical for mucins that we also have in our throat, our eyes, and so to keep our tissues moist. Also coming back to the idea of Nicole, this is very long gene, has been horizontally transferred. We do not know from which organism. It has a high GC, and there are these remnants of very low 80 or very high 80 around it, but it has all the characteristics of a horizontally transferred gene. Now, we showed that it was really extracellular, that it's outside of the cell. So it's a very large protein, and it has to be transported through the membrane, which is also a very energy costly process. So we predict that these mucins, like we can see in this picture, this is the side of Halicodratum. So you look at the flat side. And there you see this mucin being formed, but it's a protection against desiccation and a potentially also a protection against viruses. So in conclusion, hypersaline ecosystems are very rich, very biodiversity rich. They contain each domain of life, including the viruses. They are very important for understanding cellular evolution and model ecosystems for astrobiology, which is interesting for the work of Carlos. And so maybe potential questions for the panel later is, could they also be tell us how life evolved, and especially do they have a potential for extraterrestrial life or for maybe terraformation, etc., etc. And with that, I thank you.
Thank you, Hank, for your very okay. Also for Hank, you can post questions on the comments about his activity on molecular ecology and evolution of marine microbial ecosystem, on microbial maps and extremophiles from hypersaline environments. He has a broad interest in community evolution, seasonal and daily rhythm in cyanobacteria-dominated environments and in the adaptation of halophilic archaea. He applies tools like metagenomics and transcriptomics on natural ecosystem and artificial-based microbial communities. So later we will have time for discussion. Our next speaker is Dr. Rachel Morgan-Keys, Professor of Environmental Microbiology of Miami University in the United States, and she will present science and fieldwork in the McMurdo Dry Valleys, Antarctica.
Thank you very much. Okay, I think, yeah, it looks like everyone can see my screen, and can everyone hear me?
Yes.
Great. Okay. Well, thanks very much for the invitation. I really enjoyed the first two talks of my colleagues. So welcome to Miami University of Ohio. It is not Miami, Florida, which I wasn't aware of when I was offered the invitation to come and interview. So I always like to point that out to people. So we're a small university in Ohio. We train a lot of undergrads, but we also have graduate programs. And my research group focuses on polar microorganisms. And I go out to their native environment. I work in a really unique place in Antarctica, which I'll talk mostly about today, but we also bring isolates back to the laboratory. So we kind of have this nice balance between studying microbes out in their native habitat, but also being able to do more focused studies in the laboratory with a number of extremophilic organisms. So the McMurdo Dry Valleys are a unique habitat on the continent of Antarctica. So most people think of Antarctica as a massive ice sheet, but actually there are these special places within the Transantarctic Mountain, which are polar deserts. And I work specifically in the valleys of one of these within the Transantarctic Mountains, and I specifically work on several ice-covered lakes in this region. But I'm also part of a larger group called a Long-Term Ecological Research Site, or LTER. And specifically, this LTER is the McMurdo LTER, and I work with a lot of other amazing scientists. So there's a group of us that are focused on the lakes, but we also collaborate closely with other scientists that study microbes in the soils. We have a stream ecologist, and we have a glaciologist, as well as someone who studies remote sensing. So this project has been funded for over 30 years, and I provided our website at the bottom. We have a great website, which obviously has lots of details about the site, but it also has full public access to 30 years of data. So it's fully available for anyone who, is interested in using our data and designing projects with our long-term data set. So a little bit about how we get to Antarctica. So we fly on commercial airlines to the South Islands of New Zealand. And then we take a military flight from New Zealand to McMurdo Station. McMurdo Station is run by the U.S. Antarctic Program. It's the largest station in Antarctica. And the station itself is basically a spot where my field team prepares. So the station has 1,200 people during the Antarctic summer. A lot of those people are support staff that basically help us get ready to do science in remote places, basically. And this is more specifically how we get to our sites. So our sites are remote, and so we utilize helicopters to fly me and my students out to the dry valleys. And these are some images of us getting ready, flying out to the dry valleys with our gear. And there's a photo in the bottom right of what it's like to hook a helicopter on the lake. So that's standing on top of a lake, and that's me hooking some of our equipment to a helicopter. So lots of fun experiences there beyond science. So we spend a lot of time sort of just getting ready to do science in these extreme places. And here's some images on what it's like to work there. So they are ice covered, but we do have what we call liquid moats. So these are a unique ecosystem that we have liquid water on the perimeter of the lakes. And depending on how warm the summer is, these moats can be quite large and a challenge for us to cross. We typically are working in the middle of the lakes, and we need to drill through 3 to 6 meters of ice with gas-powered drills. So again, lots of other kinds of activities besides science. And then here's a couple of images down on the bottom right of us doing some sampling. So the middle one is some students measuring PAR, or light availability, through an ice hole. And then the one on the far bottom right is me actually trying to safely sample the moat. So we sometimes we also have experiments studying the unique communities in the moats, and they're kind of hazardous to get to. So this is kind of a way we designed a peristaltic pump and a long tubing with a weight at the end to sample that ecosystem. So a few really unique things about the lake. So they are one of the very last permanently ice-covered ecosystems, lakes in the world. There are some in the Arctic, but they have experienced a high level of climate change, and now those lakes are ice-free in the summertime. So the Dry Valley lakes are very unique from that point of view. They're also very pristine. There's minimal human impact or interactions besides the scientists. They're very extreme conditions, so it's a completely microbe-dominated food web. And the ice cover also has this interesting effect of a highly stratified aquatic environment. And on the right is a little cartoon of just generally what that stratification looks like. So again, there's a permanent ice cap on the lakes. The upper water column where there's light, or the photic zone, it's typically highly oligotrophic, so very low nutrients, excessively high oxygen, and then we have a permanent chemocline. And below the chemocline is often anoxic, little to no light, and some of the lakes are hypersaline. So the organisms there experience multiple stable extreme conditions, which my lab's very interested in. And then last, another fascinating thing for people that are interested in photosynthetic organisms, it's 24-hour daylight in the summer and 4 to 5 months of darkness in the wintertime. So the life is harsh there, and so life is small. Life is all microscopic. There are a number of interesting different ecosystems in the dry valleys, and my lab specifically focuses on the microbial eukaryote community, or the protists. And these are some images that a student of mine captured of the wide morphological diversity of these protists in our lakes. And so these protists or microbial eukaryotes play really outsized roles in the food web. So as I mentioned, the food web is completely microbe-based. There's not even any like aquatic invertebrates in the lakes. And so we've been working on building this microbial-based food web and understanding it for a couple of decades now. And this is kind of our current view of the food web, including these arrows are showing interactions between different microbial components of the lakes. And so the green arrows are showing positive interactions. So for example, we're studying interactions between photosynthetic protists and heterotrophic bacteria. And then the red arrows indicate negative interactions. And so for example, the top predators in our ecosystem are predatory and parasitic protists. And so they predate on heterotrophic bacteria, but there's also some interesting things like algal parasites, like this image of Personia, for example. Another interesting group we have are constitutive mixotrophs. So these are protists that are capable of photosynthesis, but they also have they're capable of predation. So they have a simplistic digestive system. And so they can both, interact positively with bacteria or negatively as predators. And just to kind of spend a couple minutes talking about the other kind of work that I do in my laboratory. So as I mentioned, we have a number of isolates. And these are two of the isolates that we've studied in my lab. So one of the things that's really a great advantage of these lakes is that, as I said, the lakes are chemically stratified. And so we've isolated organisms from the same lake, like these two that I show here, that are uniquely adapted to the layer of the lake that they were isolated from. And so these are two Chlamydomonas species. So Chlamydomonas is basically a single-celled plant. So it's a very good model for studying processes of photosynthesis and other plant processes. And we have studied this organism, Chlamydomonas priscui, for a couple of decades now, and it's really become a model of cold adaptation for photosynthesis. So it was isolated from the deep photic zone. Obviously, it's low temperature adapted, but it's also adapted to high salinity. So that's a really extreme combined stress for a photosynthetic organism to deal with. And more recently, we isolated a second Chlamydomonas, which we named Chlamydomonas ice-MDV. It is from higher in the water column. It doesn't have the same high salt tolerance, but it has a unique physiology, including a lot more plasticity in its physiological responses. And so this slide, which I don't have too much time to spend on, but it's basically summarizing a culmination of 20 years of work in Chlamydomonas priscui. And it has some really fascinating physiology, and we're still working on this organism. So some of the things that we've discovered in this organism. So on the upper left here, it says no state transitions. So state transitions is a very conserved response across all photosynthetic organisms to rapidly redistribute light energy. And our organism, C. priscui, also called UW241, totally lacks this common response, probably because it's adapted to these very permanent extreme conditions. On the other hand, it has really good tolerance to environmental stress, such as the combined stress of low temperature and high light. And so that's what this figure is showing. So on the left is a typical temperate mesophilic species, Chlorella vulgaris, under, the label got a little messed up, but under low light and high light, under low temperature stress. And you can see the organism is very stressed. It loses its chlorophyll. The Antarctic organism doesn't bother it at all. Just basically looks the same, keeps growing, can actually grow faster under that stressful condition. And down here is basically some data about the photochemistry or photosynthetic health of the organism. So basically it's showing that it keeps all its chlorophyll, it can grow faster, and this is a measurement called excitation pressure. It's under much lower excitation pressure. And one of the reasons that it's capable of this is that the organism has constitutively low reactive oxygen species. So this is a kind of a relative measure of ROS here, comparing, again, a temperate species, SAG 4972, with our Antarctic organism. And basically, regardless of stress, it has very low levels of ROS. And we've gone on to characterize this, and it has a really constitutively high activity of the ascorbate peroxidase pathway. And then in the last minute, I'll just mention the last finding that we're still characterizing today. And that's these two figures. So this one up here shows novel phosphorylation patterns. So the Antarctic organism has a completely novel example of phosphorylation. So it has underphosphorylation of some proteins, and it has overphosphorylation of novel proteins. And we think that this phosphorylation is related to the organism being able to assemble this membrane supercomplex, specifically photosystem 1. Some of these novel proteins are part of this supercomplex, and the assembly of this supercomplex allows this organism to drive high rates of an alternative electron transport pathway called cyclic electron transport. And this pathway is related to, again, constitutive stress protection and the ability to maintain high rates of ATP synthesis. So we think this is also, all of these constitutive adaptations are really important for this organism to survive high salinity and low temperature. And so to conclude, the McMurdo Dry Valleys are natural laboratories to study questions around microbial ecology. The protists play outsized roles in this ecosystem, and the MDV lakes are genomic reservoirs for novel extremophiles. So thank you very much, and thank you again for the invitation.
Thank you for your talk. Also, for Rachel, you can post questions about her experience on the adaptation of the photosynthetic apparatus in a pseudophilic green alga isolated from ice-covered lake at McMurdo Dry Valleys, and then on the bacterial fatty acid pathways in the laboratory of John Cronan, and then on the photobiology of a thermophilic green sulfur bacterium. She has been going to Antarctica for fieldwork since 2005. Thank you for your talk. And then we can move to our next speaker, Dr. Kashuri Venkateswaran from NASA Jet Propulsion Laboratory, leading ISS Microbial Observatory Project, and he will present microbes in space.
Thank you very much for the invitation. Serena, are you able to see my talk? Are you hearing me? Hello?
Yes, we can hear it correctly. Thank you.
Okay. You're seeing as well. Okay, thank you. So the previous talks were phenomenal. They took us to the deep sea, as well as high salt concentration place, as well as the cold Antarctic. And now I'm trying to take you to the space. So I'm Venkat from JPL. We have done so many work based on the, what are the kind of microbes associated to these rovers, or the, space station, what kind of microbes are there, where in a confined situation. And more than that, can we take those microbes to our advantage and then take intentionally to the space by getting some sort of space payload so that we can have a controlled experiment up in space. So, the previous speaker was telling you about how difficult to go to the Antarctica in the earth. I totally understand even going down to the deep sea, which I myself went down. It is not easy. But now let me show you how we can go to the space. So I'm not able to cover all the talk here. So one of the things that what I'm going to show you here is, can we expose microbes of our interest? That means, when you think about the space, it is biggest issue is the radiation. Can the microbes that we see that is highly, extremely resistant to this radiation thing, can we take it to the real space and then put it outside the space station and see how long it can survive? This is clearly why we need to do this kind of microbiology in NASA, because astrobiology. That means, if you're going to go and look for life in other planets, better don't take it with you. So that is one of the things we call planetary protection. We have to protect all other planet all the time if you are going to look for life in those in those places. But obviously, if you are going to the moon, which you already know there is no life, there's no issue. But if you're going to the Mars and beyond, yes, there may be chances of life there or early life in there. So, but we cannot contaminate with our own thing. That's all robotic missions. But we cannot simply keep quiet, because human mission comes in place, so that microbes are our friends. So can we utilize those things for our own purpose, okay, for a sustainable living of the human for a long period of time? There is a human mission. So human are walking fermenters. They carry all the microbes with them. So when you put human in equation, no chance that you are going to contaminate all the place. Let's see how it works. So this is the place that where I work. There's a couple of minutes of video. Then you tell how we actually control the microbial contamination that is going to Mars and beyond. 2 minutes video.
It's really important that we send a rover that's clean and we make sure that it doesn't contaminate Mars. My name is Mujige Stricker and I protect Mars from Earth bacteria. The next Mars rover is slated to go to Mars, collect samples so that eventually we can bring those samples back to Earth and determine for the very first time, did life exist on Mars? There's nothing that we can build that's sterile. So we take swabs and wipes of the spacecraft as it's being built. It gets put in an oven, it gets put in various chambers and clean rooms so that we can maintain that level of cleanliness. If we do find something on Mars, we have to make sure it's something that actually came from Mars and not something that hitched a ride. This is the place where the magic happens. Oh, it's definitely cool. In this lab, we look specifically at spores. So spores are those hardy microorganisms that can actually survive if it made it on the spacecraft in the journey through space, through the vacuum. It's very humbling to be a part of this big project because there are hundreds of people that have to come together and build a spacecraft. There's no one person that can say, I did this, I made this happen. It's always a we. I owe it all to Carl Sagan and watching the cosmos. I remember being a little kid, going to the public library and renting that VHS. And from that moment, the light bulb turned on. It actually was the start of my passion of science communication. We are citizens of our universe. We have to be good ambassadors when we are exploring other planets, other moons. So it's the right thing to do.
She was my postdoc and then a student and a postdoc, and now she's a program manager for Mars Science Return mission. Anyway, so you have given a glimpse of what we are doing in our lab. So you are collecting so many different kind of microbes that are and exposed to various different things, including the space radiation conditions. So there are so many translational benefits out there, from basic science to enhancing human health in closed system, and also pathogen detection and countermeasure, because you cannot simply identify the problem, we have to come up with a solution. So we can leverage the omics data sets for medical practice. There are so many applications that I can take off from here, but I don't have time, so I'm going to do only this number 3. Bacillus pumilasturi, this is a catchphrase, that is space to your face. That means when you are exposed to this kind of microbes to the space, that will benefit to your Mother Earth as well, because we are doing a lot of genomics, proteomics, metabolomics, and because of those things, out of the world space to your face product development was also happen. Okay, first of all, Hank was also telling you that it cost 100,000 euros for getting one genome. Same here, $100,000 we spent to get this particular whole genome in 2,000 times period, and now you can do it in $100. And first, why do we need to do this kind of things, right? First of all, we need to see are they problematic. The answer is yes. No matter what, how much you clean, whatever it is, you will find spores in such a oligotrophic environment. That is what humanly possible that we can do, and microbes are clever than that. They know how to get into these places and all. Okay, so. But what kind of spores are the problematic? So basically, Bacillus pumilus are the one always present where whatever you do. So we got the whole genome of this particular strain because this is highly resistant to UV radiation. And also, the modern day spacecrafts are not made up of heat resistant materials. For that, we are using some sort of chemicals to clean various surfaces. These extreme electronics are able to withstand those conditions, but it won't withstand the regular sterilization process that we normally do. Anyway, so these are also resistant to chemicals are being used, hydrogen peroxide in the surfaces, and also these UV 254 radiation resistant things are also resistant to simulated Martian conditions. So these are the basic studies worth of 10 plus years that we have done to convince NASA, hey, let us look at in space. Because going to space is not an easy job because it costs money and resources. So first UVC 254, which is the known DNA breaker, double bond breaker, so that the life will not continue. And the Bacillus subtilis 168 is a biodosimetry strain which American Public Health Association is using to sterilize the drinking water. Around 200 joules per meter square is good enough to kill everything. That's what the American Public Health Association proclaimed. But some of these wild wolf, the Bacillus pumilus SAF432, cannot get killed. It is only one log reduction, even at 2,000 joules per meter squared. So this is one important part. Okay, then you can also simulate the Martian conditions in the lab by putting appropriate sample holders, and these individual sample holders that you are seeing that you can put, it is 13 millimeter size, and you can put the appropriate spores in it, and then put appropriate sensors like temperature and humidity sensors to see what kind of temperature is there. Then you can also mimic to this Mars conditions and pull the vacuum out, and the Martian atmosphere is roughly 6 millibar. So you can pump in appropriate gas that is relevant to Martian conditions to keep it 6 millibar. Okay, this kind of setup, and now you can hit the xenon arc lamp, and then you can generate the 200 nanometer and above UV radiation that is normally experienced in Mars. Okay, these are the conditions that you can set up and then simulate and then expose these kind of microbes and screen what is the one that is actually surviving such conditions. Okay, so when you look into the fact, this is the dosimetry strain that which I already told, Bacillus subtilis 168 can get killed 2 minutes. So that means, people may wrongly think that Martian conditions, including the UV, basically can take care of everything. You don't need to worry about life, but that is not the case. So this kind of wild wolf that we isolated from this clean rooms, actually, while assembling this Martian even after 30 minutes. This 30 minutes is very, very crucial because when you have a lander go and land there in Mars, it actually propagates the dust. So when it comes to the dust and other things, the bugs that actually associated to the lander and the rover may get protected by those dust. Let's see about that. So I'm not having time to show that one. So it is actually higher, resistance. In other words, 1 log reduction only you will see if you have such a kind of protection. With all these things now, you can have a science payload like this. As you see, is that there are so many slots here. It is not one-man stuff. It is multiple people work together with the European Space Agency and other space agencies together, and then develop this kind of payload. It costs roughly $2 million, and the total science payload plus if you put everything together, roughly $5 million worth of resources you need. But before that, as I showed you, you need to show why you are going to take this kind of bacteria or fungi or whatever it is to the other outer space, right? So if you put a shield completely closed, that is called dark. That means you are not shining any UV penetrate through this. That means, UV is cut off completely. And some of these things that you are seeing, this actually have a Martian atmosphere, 6 millibar, which I showed you. So it's a very tightly sealed in terms of the gas leak, but it will have the Martian atmosphere, the gas. And some of them is having 110 nanometer filter, which is magnesium chloride. So that this is called space UV. And some of the other things are actually 200 nanometer can pass through, which is supracell, and that is called Martian UV. So the one it is open, but magnesium fluoride things, it is called space UV or UV space, or if it is closed like this, it is dark space. Okay, here it is tightly sealed. Martian conditions are there, so it is called Martian UV. If it is closed, this one, that is dark dark Mars. So it left 2008 and then actually planted outside the space station. This is the European Columbus space station of the International Space Station part, this Columbus module. And here is the one that our little space payload that I just showed you is implanted by the astronaut spacewalk. And here is a little bit closer look, and this is the same thing that you are seeing. So it went there in 2008 for about 18 months. We exposed outside the space station, experiencing real space conditions. Okay, and then after 18, it came back to our lab, and then we were trying to do so many different things. So this bare aluminum is not actually, it's a completely polished aluminum. You can even look your face like a mirror. But when you look under this scanning electron microscope, this has pits and falls. When you have pits and falls, these kind of microbes settle in there when you prepare this monolayer. But it sits in there, and then it gives you a kind of cushion of, from the UV radiation. So after 1.5 years, we can remove all those 100% out if it is a ground control. But the one that actually taken 6 or 7 log reduction, that is clearly showing that it is reduced from the ground control for sure. So when you look at the real, we did so many different kind of genomic characterization and all the kind of stuff. There is no genomic changes, because these are spores, metabolically inactive. That's why you are not seeing such a kind of genetic mutation. But actually, when you are doing, there are certain phenotypic difference. So this one is UV space 5062 variant of SAFAR 32, which we sent it out, which is the control here. Control get killed off around 2000 joules, but this is continuously moving and not get killed even 4000 joules. Is it due to mutation? I don't think so. It is some set of protein changes is happening. That's why it is phenotypically different. This is the first generation spores, okay? And if you're looking at the vegetative cells of those first generation, this is dark space 40T5. And as I previously showed, this is 200 joules it can kill, which is vegetative cells. But this one is spores, okay? Spores 2000 joules and vegetative cells 200 joules per meter square. But the dark space that came back from the space, it's even 900 joules, it is continuously growing. What that means that the vegetative cells is having something that is protecting it from this radiation. That's what you want to look into the metabolites, right? So the proteomics is showing that there is a differentially regulated, either upregulated or downregulated in this particular dark space compared to the all other, including the control. So this is a very good candidate for moving to see what it is, okay? With all the different proteomic analysis and the validation of various different analysis, it concluded that all taken together, the functional assays demonstrated that the activities of TCA cycles and energy metabolisms were decreased in space surviving strains. So that means, think about a marathon runner versus the 100-meter dash. So if you're asking Usman Bolt, he can run 100-meter, but he cannot run the marathon of 42 kilometer, right? So likewise, the sum of the bugs that is slowly metabolizing is actually surviving, but not the one that is reproducing quickly, that will die off. So with this, what we were able to get it, the metabolites are producing something different. We just saw the proteomics differentially expressed. So that is the product that, NASA won't do this product. We have licensed to the private company. So they develop this space to your face thing with this kind of microbe that is exposed to the outer space. And this is called Ionia. Some of you from Greece know Ionia means eternity. This age-defying serum is commercially available right now with our space technology, okay? So what is this actually? It helps to restore moisture to your skin and improve visible signs of aging such as fine lines, dark spots, dullness, and wrinkles. And it is nothing but a hyaluronic acid that any cosmetics that you have, it is there. But this space flown, bee pumilus produce certain same thing, but the activity is different. It is not injurious to the human cells, and also it allows it to, show this signs of anti-aging. So this is what Omics in Space is doing. The issues and solutions are, from environmental omics, this can be exploited to enhance human health and well-being of the closed system. The same compound that I showed you can be used for the spacesuit to attenuate the UV. So Omics in Space Research aims to translate findings in fundamental research to medical practice and meaningful health outcomes. The Omics data sets were placed in NASA Gene Lab bioinformatics environment. So anybody who are interested, they can get into this and search for this Omics data so that you can get some innovative science coming. This is not only by one person. This is the product of several postdocs and students and investigators are all over the world. Thank you very much.
Thank you, Venkat, for your talk. Of course, you can also pose questions for Venkat about his experience in field studies in several extreme environments, such as deep sea, spacecraft missions, assembly facility clean rooms, and the space environment in Earth orbit. He also provides expertise for non-NASA programs, such as commercial agencies and medical industries. The bioinformatic dataset generated by Venkat's team are extremely useful in the development of biosensors, which will provide information about the spacecraft surfaces and enclosed habitats in attempt to determine forward contamination, as well as develop countermeasures to control the problematic microbial species. So we can start with the discussion with all our speakers. We have a lot of very interesting questions. I will start with a question for Nicole. Do you think that viruses can also help bacteria in infecting eukaryotic cells?
I don't think they would help them directly, but indirectly, I think through horizontal gene transfer, as we're seeing in so many microbial environments, that the viruses can transfer genes, and the selective advantage then of the bacteria of taking up these genes in the environment that the viruses are also in will enhance the ability will provide a selective advantage, put it that way, to the microbes that are associated with eukaryote or in any other environment.
Okay, thank you. There is a question for Hank. Quite peculiar. Is there a significant reason behind the square structure of the microorganism?
And so actually, I always explain that it's not really square; it's flat. And meaning it is flat, it has an optimal surface to volume ratio, because all those processes that take place at the membrane, like the phototrophy, also oxygen uptake, all the nutrient uptake, it all takes place at the membrane. And if you have a very big spherical cell, you do not have enough time for the transport of all the solutes and all the things to all the parts of the cell. If you're completely flat, everything can be transported everywhere. In addition, if you're flat, how do you divide? So you can't really go divide a slice through a paper. It's very difficult. If you're circular, you would end up with pie charts. So I think the most logical consequence of being flat and dividing is that you end up with square corners.
Okay, we have also a question for Rachel. I'm very interested in how you prepare for such field work. Do you first training? Do you physically train to be ready for such extreme condition? How do you handle basic human needs while out in the ice? I find it fascinating that some researchers are going through this while sit in front of my laptop analyzing data and complaining about the office suboptimal temperature.
Yeah, thanks for the question. Yeah, so I do first aid wilderness training before every I get certified for that, so that the lead person on the team has like first aid training in remote conditions. It's very hard to prepare people to go there because it's like going to another planet. So but I think actually what's more important is building a good team and making sure like you sort of have good team dynamics before you go to the field. So we do a lot of hiking with packs and I do sort of a series of kind of training, PowerPoints to kind of prepare people for different parts of the field. And then I guess the second part of the question is like living out there. So I didn't have time to talk about it in detail, but we, because these sites are heavily studied for a long time, there's some primitive buildings there that help us a lot. So we actually camp. So we tend to out there, but there's solar power and there's some primitive buildings, and particularly there are lab buildings, tiny lab buildings that have power and heat, and that's actually a huge benefit versus just working out in the conditions is very, very different from being able to work in a nice camp that has some, primitive buildings and things like that. One last thing is Antarctica is a highly protected ecosystem that has the Antarctic Treaty. So part of that is leave no trace or leave no waste. So we have to be very careful about our science there, but we also have to be very careful to collect all our waste, food waste and other waste, and take all of it out of the camp with us. So that's there's a lot of logistics around kind of living and, you know, being environmentally conscientious there.
Thank you. He said, for Professor Venkat, what about the moon missions? Is it always been a standard to make sure all instruments, spaceships, and so on are sterile and don't carry microbes on the moon? Since astronauts were there in the past, how did they ensure that the human microbes were not on the outside of their suits, if they did? If not, which are the implications?
Thank you very much for this question. It's an excellent thing. So there is a forum called Committee on Space, which is called, COSPAR. So that dictates what are the requirements for the exploring planet, including moon. So moon being visited multiple times, collected samples from there, we know for sure there are no life forms that we know of today. What we know, the life is carbon based. So based on the carbon, we don't have life in the moon. So because of that, we are not having a specific requirements like the one we have it for Mars and beyond. So when I said Mars and beyond, it's not Jupiter. Jupiter is highly radiation resistant, so you don't have it. You may not have, but Jupiter moons like Europa and Saturn moons like Enceladus, all these things have some requirements. If you go and then land there, you have to clean. Now there is no word called sterile because what we know today is below detection limit of your detection system. So that means they have a recommend saying that 300 spores per meter square will not make Mars red become green. That means things will not multiply for 50 years or something because of the conditions that we know. So you have to put some requirements that is humanly possible, right? So we already decided moon has no life, so you can freely go there with the instrument and other things to develop something for other planet. In other words, if you are going to develop a closed system where human is going to live for 1 year, moon will be a stepping stone to look for it. For that, we are not going to dump things like what Rachel said. You are going to Antarctica and other things. You cannot dump things, but you clean to the extent possible, but you don't have a stringent measurement like what we have it for Mars and other planet. So that says to you that the moon material dust may get into your thing through your suit and then become a problem for yourself. So we develop an appropriate suit that is not allowing external material come inside. So for moon, there is no issue right now. The requirement is not there. Hope I have conveyed that requirement needed based on Committee on Space Research.
Okay, thank you. There are now some questions that are for all the speakers. This one, for example, Dr. Venkat and Dr. Bullis mentioned that slow metabolism is a plus for surviving extreme conditions. Is that the case for all extreme environments? I don't know who wants to start replying.
I can start at least for the sultans. It is a fact for the Halocodratum. It's a very slow growing organism, but it's not necessarily true for the other halophilic archaea in these sultans. There are some that have a doubling time of 6 hours, or they can almost compete with they cannot compete with E. coli, but they can compete with many of the regular bacteria that we know. So I can't say if that's a fact for all the ecosystems. Maybe the others can add.
Any of the other speakers want to comment about the environments there?
Ms. Venkat. So what Hank said is absolutely right. It is not universal for all the extreme environment, but some of the environment like oligotrophic by nature is nutrient limiting environment. So because of that, actually, if you dump a lot of things, it will die off basically. You take that oligotrophic microbes and grow in a nutrient-rich media, it's completely die up. But if you have a very, very low nutrients, like I think Hank also showed that after a few years, something will pop up. So we also seen that, giving paints that we are using, paint that we are using for these surfaces that is going to Mars and beyond, actually allow these bacteria will use it as a nutrient, but it takes years to grow.
Okay. Nicole and Rachel, you want to add something about that?
Yeah, I think mainly, my organisms grow pretty slow because of the thermodynamic constraints of low temperatures, basically. So, but they still do pretty well. So I think they can grow between like 30 and 80, a doubling time of 30 to 80 hours. And one of the recent things that I didn't talk about that we were really surprised is actually, even though one of our organisms is from extreme shade conditions, it can actually respond really well and grow quite a lot faster if you give it more light conditions. So we can get it up to a doubling time of about 20 hours, which we were pretty surprised that it had that capability.
Yeah, and I can say that part of the selective advantage in the oligotrophic environment of the deep sea is that, and particularly where you have reductants and oxidants, and as a chemosynthetic bacterium, you need both, that living with the host actually allows you to access both reductants and oxidants because it's bringing them to you. So their doubling times actually are quite fast because they're feeding the host. And if they're not doubling, the host is going to starve. But we can't culture them. So, and nobody has, although they've been trying for 20 years. And based on their genomes, it looks like the host has very quickly taken control of some genes that allow them to control their growth and might make it difficult for us to do it, at least in pure culture.
Okay, another very interesting question. There are many microbe conservation initiatives. How much are they focused on extreme environments? And there are ethical implications linked to them. For instance, Dr. Venkat showed that some species might have commercial applications.
Yeah, so if you are doing some research, if you wanted to see the return when you, during your lifetime, better get some biotechnologically, useful things to get done so that you feel more, interesting there. But at the same time, you cannot, you said about ethical issue and other things, that means if you are taking from the extreme places and that become problematic microbes, like if it invades into some species and then eradicate those kind of stuff, that is a problem. So we have to first make sure whether they are problematic or not, and then based on that you can do, because extreme environments have a wealth of knowledge that is useful for the humankind, and you should exploit those things to our understanding. Who knows that if you are taking some microbes, or if you're getting some microbes, or if some microbes adapt to the microgravity conditions, might produce some anti-cancer compounds. So these are positive things, not ethically issues.
Okay. For the other environments, do you have any conservation-specific initiatives or any ethical issues regarding the microbial system that you are studying?
Maybe if I can add something to this. So, not specifically for the hypersaline ecosystems, but I think in general, what we would like to do is something like biobanking, and don't only go for isolations and for isolates, because most of the majority of microorganisms still cannot be cultivated. But it is very important, especially if you go to places where sampling is very difficult, like the deep sea, like certain places. It's very easy to go to the salt lakes and stand between the flamingos. It's fun, but the deep sea, etc., is very difficult, very expensive, and we do not yet have all the techniques to characterize everything or to isolate everything. So biobanking, either in freezing at liquid nitrogen, also is a very important tool. And also using the metagenomics approach to see whether there's anything in there that may be of interest. So we have to combine these techniques and then see whether maybe in 50 years from now we can go back to these samples and say, oh, wait a minute, there's something interesting here. We now can apply it.
Okay, thank you. There are also several questions about sample handling. For example, this one, there is a required time for us to bring the collected samples in the lab. Were there are any challenges traveling the collected samples to the lab?
I can answer.
Go ahead.
Sure. Yeah, it's a huge challenge. So the U.S. Antarctic program has an amazing infrastructure in place for shipping samples, which is very important because the samples are really precious, and there is a lot of taxpayer dollars basically invested in collecting those samples. So they have an extremely good system. But I do run into difficulty because one of the most difficult samples to bring back is cold, not frozen. Basically, it needs to stay at 4 degrees, but it cannot freeze. So basically live samples. And that is a very big challenge for me to bring back, like live organisms, to cultivate new things. Or sometimes we're interested in bringing like bulk lake water back with live organisms. And so I've run into a number of challenges with particularly those kinds of samples. And it's also really important to bring those samples back because it lets like more students experience working with, microbes from and samples from very, remote places that most students aren't going to get to experience. So it also increases students' ability to, interact with those samples. So it's really important, and it is challenging. It continues to be challenging.
Yeah, maybe our challenges are, the first is that we ideally want to fix our samples in the deep sea. So we've developed some nice approaches where we've brought RNA later, the fixative for fixing both DNA and RNA down to the deep sea, and pilots have to manipulate with the ROV arm, with the submersible arm, to actually crush the mussels just so that they don't damage them, and throw them in RNA later and bring them up to the ship. And we're comparing their expression patterns to those that we fix on the ship. But then the next challenge that we face, and that's been very interesting because it looks like the mussels don't care so much, but actually the bacteria go into anaerobic metabolism, and their metabolism is totally different. Their transcriptome when we fix them in the deep sea. But then the next challenge is that after we've been at sea for 5 to 6 weeks and collected all these precious specimens, is that we often go to ports that are quite small, and then we don't have NASA behind us or other organizations that help us ship it to our labs. And it's a real challenge to find companies that are specialized in transporting samples from very remote places on ice. Same as Rachel also said, that some have to be frozen, some have to stay at 4 degrees, and we pay a lot of money to, I guess I shouldn't advertise, but to a company that does a good job. And then finally, also maybe the challenge that many of us are facing is the Nagoya Protocol, because it was developed for many good reasons, developed against exploitation of resources of countries that might not have the access that they'd like to, but the paperwork is a nightmare. So that those are the challenges we face.
Okay. Venkat and Denk, you want to add something about that?
Yeah, bringing samples from space is a big issue as well, and you have limited space when you bring it down. So that is one of the things you, as a researcher, you want as much as samples possible, but there is a space constraint that comes below from there. So there is a huge issue, and also how you are going to bring those things under cold conditions, or frozen conditions, or regular temperature conditions. All those things matters thing. And Nicole has said about the costing money. So that is the thing we call them as feasibility study. So they'll say, you are dreaming big, Venkat, but we cannot do it because only we have this much resource. So,
Yeah, I can only add to what the others said. It's whatever you bring home, the samples that you take from the field into the lab, it will change. So the community will change, the species will change somehow. You have this lab evolution. So there is no perfect way to treat the cells, and maybe the best methods to fix them or to freeze them may again be very detrimental to the organism. So in the end, it's not difficult to bring them home from a salt lake. You can just have them in your luggage. There's no special needs there, at least from the nicer tropical solar saltants that I work in.
Okay. There is another question about ethical concerns. As we study and use microbes from extreme environments for things like bioremediation, making energy, or space mining, as we heard in the previous session, what ethical issues should we think about? For example, how we can avoid harming ecosystem biodiversity or causing unexpected problems when using the microbes in new ways?
For the deep sea, I can say that we developed a code of conduct quite a while ago, 10, 15 years ago, where researchers got together and said, what do we think is appropriate behavior in the deep sea? Can we, how can we protect these very special environments and ensure that our sampling isn't a problem? For example, and when they were first discovered, researchers were taking symbiotic organisms like the tube worms and transplanting them somewhere else to see, how they do at another site. So we no longer do any of that. We're very careful. We try not to sample the chimneys, or only if by accident the ROV, the submersible, somehow or other damages them. So I think we're very, very conscientious about that. And I think the other side is a political question. For example, whether mining in the deep sea is any better than mining on the land. That's a whole nother question that luckily, how should I say it, we can be active, but as biologists, that's, in my opinion, a question that the public, the companies, our expression, and the politicians need to answer.
So that's for the very reason we have a requirement called planetary protection requirement that is agreed by the UN treaty by all the spacefaring nations, whether it is Russia or India or China, anybody, they meet once in 2 years, and then they show what they are doing and whether they are following this UN treaty or not. So that's why that particular gentle human agreement is up there. But nobody is policing one country to the other one. It is up to you what you are doing. But this space mining and other things are still not there. First of all, we are trying to do in situ resource utilization, which is different from space mining. In situ resource utilization is nothing but you cannot take the soil from here to build a habitat or build a house in there. can you make concrete with this, with the soil that you have it there, with the biological product? It is called biocement, that can be used to build a habitat, like a igloo box that you can think about. Okay, so these are the things is in situ resource utilization with the help of the microbes. For example, we are also trying to generate plant, the food, right? So how whether plant can be grown in other planet or not, everything is in a contained area. It is not simply we are making red planet green right away, but there are certain group wanted to do terraforming, which is not agreed upon by majority of the people, but there are some group wants to do, it is called terraforming group, which they don't care, they wanted to grow something and all. So that is not there yet, it is still in the paper one.
Okay, if there is any other comment about that, I want to acknowledge Nicole, who has to leave us a bit earlier. Thank you for your participation and for your interesting contribution, Nicole. I hope we will have time to discuss further your research in the future. Thank you for participating and goodbye.
Yes, and my apologies for having to leave early, but the German train system is a wreck. Used to be the pride of Germany, but that's another discussion. So thank you, everyone. I very much enjoyed this, enjoyed your questions, and see you another time. Bye.
Bye.
Thank you, guys. Bye-bye.
Bye.
No, it was only for Nicole. I think we have still some time for discussion. I'm missing Venkat
Yes,
now.
yes. I'm sorry. Yeah, I think there is some issues backstage. We have connection issues, and our team is all over the world, and it's difficult to communicate behind the scenes. So no worries. Yes, we still have time. My apologies.
Okay. Another question almost on the same topic. Could introducing extremophiles in new environments outcompete native microbes and alter ecological balances? How can we assess and mitigate this risk?
So by rule, you cannot introduce anything to a new system. You can do it in a controlled way in the lab or anywhere, but you cannot simply introduce something into a new environment. That is a big no, basically. Go ahead, Hank.
Yeah, I think it also has to be clear that an extremophile, the name says is filed, it loves the extreme conditions. So if you introduce my halophiles, like the Haloquadratum to the sea, it will immediately have an osmotic shock and blow up and explode. There's no survival there. So it may only work if you go from one extreme environment to exactly the same other extreme environment where the organism isn't yet. But if that happens, indeed, like Venkat says, the chance of survival is small, and well, you probably need to introduce a lot of organisms before it makes some sense to take over the population. I mean, it can happen. It can carry a virus. It can. You can't exclude things, but I wouldn't be too concerned.
Yeah, same. There's very strict guidelines in Antarctica. You cannot introduce non-native species there, and I have had the permission to transplant communities under very controlled conditions between lakes, for example, to mimic some like mixing events. But we have to basically convince them that we're not going to, we're going to mitigate an environmental release.
With that respect, may I ask Rachel a question?
Sure.
I mean, there was always this big concern when they start drilling this deep sea or the through the deep ice and these Lake Vostok, I think it was, there were always these concerns of contamination and ecological upset.
Mm-hmm.
Is there any follow-up studies of whatever happened to the system? Was there an ecological problem?
Yeah, so there's kind of two different kinds of groups working on subglacial lakes. So the Lake Vostok is mainly the Russian science groups, and so there's not very good information, on their, especially more recently, I guess. But so I do know that I had colleagues that visited Vostok and were, part of consultations and things, and they were quite concerned about the way the drilling was being done there, and drill fluids not being well, monitored and things like that. The other type of science is the some U.S. groups. So there's been, and also the British Antarctic group as well, but I'm more familiar with the U.S. group. So they've drilled 2 subglacial lakes, and they use a hot water drill. So they basically use sterile hot water, and and also they have like a UV sleeve. So they have pretty high stringent, rules in place to really prevent contamination of those subglacial lakes, which are on a whole other level of pristine compared to the lakes that I work on.
Okay, thank you.
Okay, now we have some specific question for our speakers. So, for Hank, sorry. Okay. For the light dark transcriptome, were these experiments performed under optimal lab condition as opposite condition closer to the native environment?
Yeah, I guess there are no optimal lab conditions. I mean, we can never mimic the natural environment. I try to do that with coastal microbial mats, to grow them in sand and in the layers and having light only from the top, etc., etc. Here we did not have the ability to completely mimic the natural situation. So yes, we always have to take into account that whatever you do in the lab is not necessarily representative for what happens in nature. May I ask, was there also time to ask questions for the first 2 speakers, or is that different session?
I think we have time. I don't know if they are still here, because Luca wrote in the chat that he has to leave.
Okay.
I don't know. Okay.
I promise I'm jumping in for the last time.
No
as
worry.
a reminder for everyone, there is a Q&A available on Cassini. I can share the link in the chat that you can do. It's chat-based, and it's open for 48 hours. Okay. So everyone can ask questions to the speakers, and they receive an email and they can answer directly to you. So thank you, Hank, for letting me say this.
Okay. Grazie.
To the discussion.
Okay. Now, a question for Rachel. What is the most optimized sampling method do you usually use to make sure that the isolate microorganisms greatly represent the microorganism in each zeta?
Yeah, that's very hard. So we have had a main interest in photosynthetic organisms. So when we try to isolate things, we use a variety of different strengths, basically, of growth media, because our organisms also live in extreme oligotrophic conditions. And so if you just kind of add some nutrients, basically you'll get a bunch of weeds like grow really great in the high nutrients. So what we have done in the past is I basically kind of start things growing in Antarctica a little bit. I think that's really important in a variety of different kinds of media. We've also done like salt concentration, like a gradient of salts and things like that, and just kind of get them going to a really low density, and then ship them at 4 degrees and do not freeze. Hopefully they survive. And when they get here, we do dilution to extinction culturing. So we basically take those baby cultures and dilute them out, across well plates to hopefully, isolate some novel things by diluting them out. But we have a big problem with sort of getting the same organisms over and over again because they're just really competitive, and they're not necessarily representing the dominant organisms. They're just, competitive when you put them in the lab, basically.
Which is a nice comparison to my sampling method that I took for the salters. Maybe you saw it on the picture, but it was a kitchen pan mounted on a broomstick.
Nice.
But it was clean and sterilized in between sampling.
Okay, another question for Rachel. You have mentioned earlier from your figure showing the relationship was positive and negative. I was wondering what type of specific positive and negative interaction, aside from predation and parasitism, do they have? Thank you.
Yeah, so positive interactions are mainly we're interested in things like mutualism, and especially again, phytoplankton interacting with bacteria. So we are interested in whether the Antarctic phytoplankton have a phycosphere, and if they recruit certain bacteria. The phycosphere, if you don't know, is this microenvironment around an algal cell. Other negative interactions, mainly competition, because it's especially competition for nutrients is the other main, I guess, negative interaction that we're interested in.
Okay, thank you. We have, another question for Hank. So, we saw during the contributed talks that also Dr. Alaya Munoz did some environmental metatranscriptomic. Do you think that in vitro transcriptome analysis for such strain still holds value? Should we just rely on in situ transcriptomics?
Well, that really depends on the questions that you ask. I mean, we just have to be aware that whatever you do in the lab is not necessarily representative of what happens in nature. That is the main issue. But if you have very specific questions like a response to light and dark, or a response to how light's high salt, low salt, under controlled conditions for only one strain, or when you even add, say, a second strain, so when you're looking at competition, this is what you can do under very controlled conditions in the lab. So there is the in vitro versus in vivo. It makes a distinction, and it really depends on the question that you want to answer. So both are equally valuable. Just don't automatically translate one to the other.
Okay, thank you so much. As we are very close to the end of this session, I want to ask you for a last comment about your idea on possible future perspective or research in extreme environments and its future potential and its applications. If you can conclude with a comment about that.
Well, I guess very specifically for my ecosystem, I think you were asking kind of about the next frontier. I think the next frontier for my ecosystem is definitely moving more into omics approaches because we're still pretty early with things like meta-transcriptomics or metagenomics. And currently, the other major area is that winter processes are kind of a black box because it's logistically really difficult it's basically impossible to be there during the winter time. So that is something I think a lot of researchers are interested in, is winter processes and being able to do research there during the winter.
Yeah, for me, I mean, although it was great and it gave me my 50 minutes of world fame to isolate Haliquadratum, I think isolation of a single species and characterizing is nice, it's good, but I think there's more knowledge to be gained from looking at the complex communities, because organisms don't live in a pure culture. These microbes, they live always in association with many other species. Also looking at the different environmental variations that you have, the day, the night, shifts in temperature, shifts in depth, oxygen profiles. I also work with these microbial mats, like I said, where you have layered structures, where one species is on top of another species, and it comes also with gradients of oxygen and sulfide and so on. So looking at how species grow and evolve also in a complex community is going to be way more interesting to study. And for that, yes, you need omics, but also good techniques and approach to measure all the metabolic, the exchange of metabolites and so on.
Okay, thank you. Unfortunately, our time for discussion is over. I have to thank you all the speakers of this session for their interesting contribution with their presentation and in the discussion. I want to acknowledge also the attendees and the organization of Microbiome Virtual Forum and Serena and all the other organizations for giving me this opportunity to engage in this very interesting discussion. And I leave the floor to Serena for a conclusion.
I joined I joined Fabiana in thanking the speakers once again. It was very nice having you today. I think the discussion was incredible. Fabiana, thank you for your sharing.
Thank you.
And I think you did a great job today. Thank you so much. So we can now move on with the award announcement. So MVIF 36 winners are, so the best micro talk has been assigned to Patsi Tastiana Fitri from the Bandung Institute of Technology in Indonesia for her talk, Gut Microbiome Diversity Analysis of Indonesian Healthy and Stunted Children. So congratulations, Patsi. And the best selected talk goes to Maria Kardakova from the University of Surrey in UK for her talk From Microbes to Mind and Actions, Uncovering the Gut-Brain Connection in Major Depressive Disorder and its implication for human health and lifestyle. So congratulations to our winners today. And it is our pleasure to have Maria with us today. So if Maria can join me on the floor here. Hi, Maria.
Hi.
See you.
So nice to meet you. Thank you so much. It's an honor for me.
It's an honor for us to have you here. So in the perfect MVIF tradition, I would like to ask you, as every study encounters challenges, which are the challenges that you encountered during the study you presented here that also granted you the award? And how did you overcome them?
So I guess I can name technical challenges, of course. It's their lack of synchronization across their bio communities and libraries, and the way we are naming our taxa, and the way we are not stratifying taxas enough. I spoke about this during my presentation as well, that we need better stratifications, we need species level libraries, etc. And probably on the personal level is that it's this amazing paper that we are currently preparing for publication and for submission. It's synchronized with my own thesis submission, and I have my defense in 2 days. So yeah, it was also a big challenge for me to synchronize everything at once. Yeah.
So first of all, best of luck for your defense in 2 days and for your submission of your work. So you already mentioned a bit what is in the next steps for your work. So, and especially for this work that you presented, are you continuing this line of research? Do you have anything coming up next?
Yeah, so the next upcoming paper would be on diabetes and comorbidities and the clustering analysis of different taxa and the analysis of metabolites of their bacteria that can be aligned with their like comorbidities progression. And the other paper would be on the longitudinal analysis on microbiome. Which is quite exciting because we're stepping into prediction model on the various health complications, because we have about 40 phenotypes to analyze. So it is quite exciting. So we can look onto the comorbidities and to move into AI era, I'd say. But I also looking forward for studies that will be published by Twins UK, of course, in King's College, and Irene Siegel's lab, 10,000 samples. We're all looking forward for this data, and maybe, I don't know, I meet colleagues here in UK in Wellcome Trust. So yeah, a lot of exciting news upcoming. Yeah.
So I wish you all the best for all of these great works that you are already mentioning. I hope to hear from you again at MVIF for your upcoming studies.
Yeah.
You can share with us your early findings as well, if you like. We'll be happy to host you again. And again, best of luck for your defense. And
Yes.
I see you soon, I hope.
Yeah, see you soon. Let's stay in touch. Thank you. Good luck, everyone.
Thank you, Maria. And okay, so it was nice to have Maria joining us today. And before finishing the session, I hope you enjoyed it today, our special event. As usual, they have been recorded. They will be available not only on the usual MVIF YouTube channel, as you may know by now. We also host recordings on Cassini, so the same platform you use for registering. There you can also find more information about the speakers, the related works, chaptering of the talks. So if you skipped or missed some specific parts, you can also go back to them. And also, as mentioned in the chat before, you can also ask questions to the speakers, or our speakers accepted to join the Q&A for the next 48 hours. So feel free to ask them any questions you may have. Then, of course, remember to vote for your favorite talk that was shared at the beginning. So today's contributed talks. Winners will be announced at MVIF 38, that will be on April 14 or 15 for Pacific time zones, and on April 16 for Atlantic ones. You can check your local date and time as usual on our website, and this will be a special event focusing on microbiome and citizen science. You can see here the lineup. We will have Dr. Michaela Borton from Colorado State University, USA. We will have Dr. Jerome Waldespoel from McGill University in Canada, Dr. Daniel McDonald from UCSD, USA, and Dr. Sarah Anna from University of Antwerp in Belgium. We are finalizing our chairs for this event. Already confirmed for the Pacific event will be Dr. Jack Gilbert from UCSD USA. And we will be announcing the chair for the Atlantic session soon. Also, in this case, all the information available on the NVIF website and the Cassini event. In May, instead, we will have another special event focusing on the role of the Nagoya Protocol in protecting microbial diversity through fair use, which was also mentioned today by Nicole Dublier during the discussion. Our confirmed speakers for this event are Dr. Amber Hartmann-Scholz from the Leibniz Institute DSMZ Germany, Dr. Manuela da Silva from the Biobanco de Biodiversidade de Saúde de Fiocruz, Brazil, sorry for my pronunciation, Dr. Benedetta Turchetti, University of Perugia, Italy, and Dr. Valentina Veneroso, also from Italy. I hope to see you next month at NVIF again. And thank you so much for attending. Keep in touch in the meantime through our usual socials. Thank you so much.